Mendel's experiments we have already discussed. The first step is definitely selection of the parents. And you know the first rule of Mendel's experiment that he always selected pure parents. Step number 2, emasculation of the female parent. Now to avoid self-pollination, he has performed emasculation on the female parent. As you can see in the given picture, the purple flower is a female parent and the plant with white flower is a male parent. So the purple flower is going to get emasculated, and in the third step, crossing to obtain the F1 generation. So pollen from a white flower are getting transferred to the stigma of the purple flower, and the first generation, it is called as the F1 generation, is obtained, and the offsprings are grown again for the next step. Step number 4, F1 is self to obtain the F2 generation. Now here you don't have to do any emasculation because the F1 generation, the purple flower ones, the pollens are now allowed to do selfing, and the next generation result will be called as the F2 generation. Now we need to observe the F2 generation and give the result and the conclusions. So these are the steps of the experiments done by Gregor Johann Mendel. We are going to start with the first type of cross performed by Mendel, which is called as the monohybrid cross. Now monohybrid means only one character is selected. Now how to write? Mendel had a very special way of writing his experiments. Now this side of the screen you can see is the way he wrote and is the way you are also going to write in your exams. Now this side is only for our understanding, better understanding. Now here first step is always to write the parents, and parents have to write information for their phenotype as well as their genotype. And phenotypically both the parents are contrast. One parent is tall, the other parent is dwarf. Genotypically both are pure, that is why capital T and small t. In the other side also you can see the representation as the same. Now meiosis will happen, gametes will be produced, and gametes will only have one of the allele. Gametes cannot have two alleles for the same gene because gametes are haploid. Now he also performed the emasculation, that is removal of the anther from the female plant. In this case tall plant is the female. And step number 3 is crossing to obtain the F1. Now you can see the F1 generation, it is heterozygous tall. Now phenotypically this one is only tall, but genotypically it is heterozygous tall. Here comes the first law of Mendel, that is law of dominance. We will follow with the statement in the later part of the lecture. Now next step of the experiment is F1 selfing. Now the F1 generation, capital T small t, is to be allowed for self-pollination, and the next generation will be the F2 generation. So following the same steps, the gametes formed here. Now here it is going to make, each parent is going to make two types of gametes, capital T and small t. Now we will also make a checkerboard. So he made a special checkerboard जिसमें आप देख सकते हो कि एक तरफ मेल के गेमीट्स है, एक तरफ फीमेल के गेमीट्स हैं. दोनों गेमीट्स चेकरबोर्ड में लिखे हुए हैं और देखते हैं कि पहला कॉम्बिनेशन क्या आएगा. सो पहला कॉम्बिनेशन आएगा कैपिटल टी, अगर वो फीमेल के उसी ओव्यूल के अंदर फर्टिलाइज करता है जहाँ पे कैपिटल टी का अलील है, तो इट इज गोइंग टू बी कैपिटल टी एंड कैपिटल टी. सेकेंड कैपिटल टी एंड स्मॉल टी. थर्ड कॉम्बिनेशन ये हो सकता है कैपिटल टी स्मॉल टी, एंड लास्ट कॉम्बिनेशन ये हो सकता है स्मॉल टी एंड स्मॉल टी. और होमोजाइगस टॉल, हेटेरोजाइगस टॉल, हेटेरोजाइगस टॉल एंड होमोजाइगस ड्वार्फ. देखते हैं इसका फिनोटाइपिक रेशियो क्या आ गया. द फिनोटाइपिक रेशियो इज 3 टॉल इज टू 1 ड्वार्फ. ध्यान से देखना, ओनली फिनोटाइपिक. ना जीनोटिपिकली देख सकते हैं आप, 1 प्योर टॉल है, 2 हाइब्रिड टॉल्स है और 1 प्योर ड्वार्फ है यहाँ पर. ठीक है, तो जीनोटिपिकली इनका रेशियो है 1:2:1.